Hello, my name is Carol Leibel, and today we want to examine the structure of cell membranes. The cell membrane is a very dynamic and intricate structure, and it regulates the material that's transported across the membrane, what gets into the cell and what gets out of the cell. The membrane is said to be selectively permeable or semi-permeable, meaning that only certain molecules can cross the membrane and others cannot. The one thing I also want to emphasize to you is that as we talk about the plasma membrane, if you examine organelles like the mitochondrion, the chloroplast, the Golgi apparatus, the nucleus, those organelles also have membranes, and the basic structure of the membrane also applies to those organelles. The differences will occur in what type of proteins and what type of phospholipids are found within those organelles. So here's a little video that talks about semi-permeable membranes or selectively permeable.
The plasma membrane is selectively permeable. It lets some substances through and not others. The hydrophobic interior of the bilayer is one reason membranes are selectively permeable. Hydrophobic molecules, those that are soluble in lipids, can easily pass through the membrane. In addition, small molecules like O2 can sneak between the phospholipids of the membrane. On the other hand, large hydrophilic molecules like glucose and ions such as sodium ions and hydrogen ions cannot pass through the membrane unaided.
So let's examine what a phospholipid is composed of. First off, we know that a phospholipid has a glycerol backbone, and attached to it are two fatty acids, and those fatty acids have hydrocarbon tails. The hydrocarbon tails can be saturated, or as we see right here, they can be unsaturated. The third component of the phospholipid is a charged phosphate head. As you can see, there are other things in that phosphate head besides the charged phosphate group. So the tails are going to be hydrophobic. They dislike water. They dissolve nicely in a lipid. The phosphate head is said to be hydrophilic, and because it has a charge to it, it is attracted to water. So phospholipids have hydrophilic heads and hydrophobic tails. When you're looking at a plasma membrane or a membrane in general, then you're not going to see all those wonderful carbon, hydrogen, and oxygen and phosphate atoms. Instead, it's illustrated like you see here. Here's the charged phosphate head, and here are those two hydrocarbon tails. Phosphate head, hydrophilic, hydrocarbon tails, hydrophobic. Now, inside and outside of the cell, there's water. And the hydrocarbon tails dislike or are not attracted to water. So in forming a membrane, you actually need to have two layers of these phospholipids. And they're arranged in such a way that the heads are attracted to the water inside and outside the cell, and the tails face one another so that they are away from the water. So this is called a bilayer of phospholipids. Now, phospholipids have what we call lateral movement. They have the ability to move sideways, if you will. If you're looking at this little diagram, it says that it has the ability to move 10 to the 7 times per second. The ability, though, for a phospholipid to make a U-turn or flip-flop, as they say, is very rare. It occurs perhaps maybe once a month. Here's some evidence for this lateral movement. If you take a mouse cell and a human cell and you mark the phospholipids with dyes, different colors of dyes, and you fuse those two cells, within an hour when you return, you'll find that that fused cell, all the phospholipids now are uniformly mixed. we want to examine what it means to have a saturated versus unsaturated phospholipid in the plasma membrane and those implications. If it's an unsaturated phospholipid, it means that those tails have little kinks in them, and those little kinks causes the phospholipids to be a little further apart from one another. And when things are a little further apart, they're more fluid, if you will. If you have a membrane in which you have a lot of saturated phospholipids, it allows you to pack those phospholipids very close together. And when they're packaged more closely together, they become more viscous or less fluid. That's because the distance has been decreased between the phospholipids. So this is right here, unsaturated. This is saturated. More fluid, less fluid. With organisms that have to face the winter, one of the things they have discovered is as winter is coming, the animals and the plants will substitute or put more unsaturated phospholipids in their cell membrane. So that's kind of interesting. A word about cholesterol. Now, cholesterol can be found in the membranes of animal cells, but not plant cells. Remember that cholesterol has 4 rings, and it also is a lipid. So it can be found quite nicely in this hydrocarbon, in between these hydrocarbon tails. And it can affect the fluidity of the membrane, but it depends upon the membrane. One of the things that is noted is that it can weakly bind to the hydrocarbon tails, and that makes it more difficult for smaller molecules to pass through the membrane. So it's like a little barrier for smaller molecules that probably would have been able to get through before. Now, here are the 2 opposing statements, so listen very carefully. If the phospholipids are saturated, by inserting these cholesterol molecules, it actually prevents them from being packed too closely together, and that's going to make the membrane more fluid. However, if the phospholipids are unsaturated, remember we have those kinks in the tail? Well, the cholesterol molecules can fill in those spaces and anchor those phospholipids and actually do just the opposite. It's going to make the membrane less fluid. So whether the cholesterol makes the membrane more fluid or less fluid depends upon whether you're talking about saturated phospholipids or unsaturated phospholipids. So let's talk about those proteins. The proteins are inserted into the membrane layer. There are some proteins that are just on the surface. If the proteins are just on the surface, they're said to be an extrinsic protein. If the protein is inserted into the membrane, then it's said to be an intrinsic protein. Different proteins have different functions in the membrane. One function of the proteins is as permeases or transport molecules across the membrane. It helps to bring molecules across. In this case here's an example of facilitated transport. Here's an example of active transport. If you're looking at that little diagram right there, those are proteins in which we see the active site of the protein, which is an enzyme. And so now these proteins stuck in that membrane are going to speed up a chemical reaction. Other proteins are used as receptor sites so that signal molecules can attach and some sort of communication is facilitated in the cell. So we have a couple of little videos and we have some other functions. So let's look. The first little video we have right here is just a simulation of the fluidity and of a plasma membrane. The blue structure is a transport protein, And then this is an aquaporin. This is a water channel that's allowing water into the cell. Alright, so moving on about the different functions of membrane proteins. We talked about enzymes, we talked about receptor sites. We also know that some of these proteins are used in cell-to-cell recognition. Also, other proteins are used to join cells together. So here we have two proteins joining two cells together, and that other proteins are used as anchors to attach the cytoskeleton and the extracellular matrix to the plasma membrane. So we call the model of the plasma membrane the fluid mosaic model, and we gave you evidence about why it's fluid. That means that these phospholipids have lateral movement and are able to move from one side of the cell to the other side of the cell. The mosaic part refers to the proteins that are inserted into the plasma membrane. Now, the evidence that we have for that is that we have the ability to use using an electron microscope to actually look inside the phosphobilayer. We've been able to fracture this membrane and to basically peel it back. And when we do that with an electron microscope, we can actually see these proteins inside the middle of the phosphobilayer, indicating that it is indeed kind of like a mosaic. Now, just a note about these proteins. Remember, proteins are made out of amino acids. Those amino acids have polar and nonpolar R groups and charged R groups. The proteins are folded in such a way that the nonpolar R groups are down here in the middle with the hydrophobic region of the membrane. And then over here on the surface of that protein, you're going to find the hydrophilic or the nonpolar and charged R groups. So that's kind of cool. Makes sense. So how do you control, or how do you make a membrane grow, or how do you decrease the size? Well, all of that is done with the interaction of vesicles. Remember, we mentioned before how sometimes there are molecules, proteins that are made on the endoplasmic reticulum, and then these little vesicles transport it to the cis face of the Golgi. These molecules move from compartment to compartment, being modified, and then we have the transface of the Golgi, which now we pinch off a vesicle, and which we have wrapped in there some molecule, and now this little vesicle is going to fuse with the membrane, and these molecules are going to be excreted. That process is called exocytosis. Now, sometimes, in addition to having these little molecules that are going to be excreted, sometimes there are little molecules inside that vesicle membrane. And look what happens. Once it fuses, those little proteins are actually now a part of the plasma membrane. This process of exocytosis is going to increase the surface area of the plasma membrane. Just the opposite of exocytosis is endocytosis. And so when endocytosis takes place, what's going to happen is a vesicle is being formed, and now it's going to come into the cell. That's going to decrease the surface area of the plasma membrane. So it's the interaction of these vesicles and endo and exocytosis that's going to increase and decrease the size of the plasma membrane. Here's a wonderful graphic of the plasma membrane. I want you to take a minute and I want you to examine it and tell me whether it's the plasma membrane of a plant cell or of an animal cell. I hope you determined that this is indeed an animal cell. And the reason for that is that out here we have the extracellular matrix, and that the extracellular matrix is found only in animal cells. In addition to that, we have cholesterol. Again, cholesterol is only found in the membrane of animal cells. We can see down here, here's the anchoring of the cytoskeleton. And here are different kinds of proteins inserted in the membrane. And so this is indeed a plasma membrane. I want you to remember that if you were looking at a mitochondrial membrane or a chloroplast membrane, that the bilayer would still be there. However, what kinds of proteins are inserted into the membrane is what's going to differentiate it from the plasma membrane or from different kinds of organelles. Alright, so that's the end of our screencast about cell membranes, and I hope you enjoyed it. So I'll talk to you later, and you have a great day. Take care. Bye.